All right. Thank you, everyone. Thank you for this opportunity, Dr. Claus. Good morning. I'm Vishal Sivasankar, and I'm a research scientist at Zia Lab at University of Missouri. And I'm here to briefly talk about building in silico minimal cell, especially about our modeling efforts and the need for experimental data. Typically, models of cell processes are robustly into disparate limits. Structural biology, which models with atomic resolution but evolves dynamics only for mere nanoseconds, or systems biology, which focuses on entire cells evolving over minutes but abstracts away space entirely. But there is a rich frontier in between these two limits where many cellular processes operate, and this is the regime of colloid physics. We are interested in biological processes inside cells that require colloid physics to explain them. For many reasons, the minimal cell is a model system to understand the fundamental process of cells. But we start with E. coli purely because it is very well studied biologically and biochemically. So now we want to pick a model process, one that exhibits rich colloidal scale interactions and is critical for cell function, one that is very well studied and seen across cell types. One such process is mRNA translation, and we choose that as our model process, and we study it through dynamic simulations. When we scrape through the literature and look at E. coli, we see that it synthesizes the majority of its dry mass as protein every cell doubling. Thus, cells that grow faster produce proteins faster, utilizing the well-known translation machinery of ribosome, translating mRNA to incorporate ternary complexes that carry amino acids. But we found that the mere increase in the number of ribosomes in these faster growing cells is not enough to explain the faster synthesis. Somehow, the productivity of individual ribosomes must increase. So we asked, how do ribosomes become more productive at faster growth rates? Now let's take a look at this beautiful cartoon showing mRNA translation inside the ribosome. Looking outside, it seems like the ternary complexes appear at exactly the right time in exactly the right order. Now, that's clearly an idealistic model of how ternary complexes find a cognate ribosome. Indeed, 30% of this central life process is mechanistically unexplained. We hypothesize that transport and combinatorics must take some time. So outside the ribosome, there is a very well characterized process synthesizing the key translation molecule, that is the ternary complex, which carries an amino acid to the ribosome. Once inside the ribosome, the process is very well understood. What is not well understood is how these ternary complexes get to the matching ribosome. We focus on these active translation molecules, and now we need a basic physiochemical model to capture how these ternary complexes search the cytoplasm for the ribosome. We explicitly represent these translation molecules as physical objects. We leverage the fact that to leading order, colloids interact aerodynamically and entropically as spheres. Here is a cartoon that explains the physical problem here. The ternary complex must search a crowded cytoplasm and compete with each other to react at a matching ribosome. This process is often overlooked because it's assumed that Brownian motion is so fast that the transport is irrelevant, meaning crowding in the cytoplasm and the combinatoric competition doesn't really matter. We tested that hypothesis. The combinatorics comes from the fact that there are 42 unique ternary complexes in E. coli, and as the mRNA indexes through the ribosome, it calls for a specific ternary complex, a cognate match. So essentially, the ribosome only sees three distinguishable particles here. Either it's a cognate match or a non-cognate mismatch or a near cognate and almost match. So with this, we constructed a spatially resolved chemical and physical framework to model this Brownian search and the combinatoric matching process. And this is how our translation voxel looks like. Let's see how this works. We first track the transport time, reaction time, which includes the mismatches and the matches. Altogether, they make up the elongation time. The Brownian motion in our simulation emerges from the first principle rather than an experimental fit. The chemical reactions on the other hand comes from well established in vitro measurements. We use these in vitro kinetic measurements to represent reaction latencies. Once inside the ribosomes, the kinetics alone can sufficiently model and explain elongation. But outside the ribosome, where we are focused, transport and combinatorics couple in the cytoplasm to set the elongation rate. So, in order to build our model, we gathered and analyzed well-established experimental data for cell mass, volume, and the sizes and abundances of the various translation molecules in our cell across growth rates. And this is where we need the helping hand from our experimental community. There has been enormous research into E. coli that helped us shape our E. coli model building. However, we are not quite there with SYN3A, and one of our major goals is to construct a database of stoichiometric and physical makeup of the cell and construct a physically resolved model of the minimal cell to answer fundamental biological questions. So let's get back to our results. We find that faster growing cells are more crowded, but the relative abundances of biomolecules in E. coli is not uniform across growth rates. The takeaway here is that the chemistry and colloid physics are coupled, and this stoichiometric crowding can explain the speed up of protein synthesis. Now that we have all our data and build our model, let's look into the dynamics. We systematically constructed translation voxels from very simple to more biologically faithful forms and analyzed expected transport, reaction, and elongation latencies by simulation. We find that as soon as there is non-zero crowding in cytoplasm, physics dominate. And this is consistently seen as we increase the complexity of our models. The takeaway here is that transport latency plays a dominant mechanistic role in regulating the speed of translation elongation, contrary to prior belief. How does a stoichiometric crowding result in a speed up? It essentially produces a trade-off between a shorter transport distance for the ternary complex due to proximity versus slower mobility due to increased viscosity of the cytoplasm, the crowding. When comparing these two, we observe that the proximity wins, which results in a faster transport. And when we compare the transport and the reaction latency, it is obvious that transport latency is the rate limiting process, thus setting the mRNA elongation rate. Okay, let's compare our findings to the experiments. Most of you might now be looking at the quantitative gap and think, okay, maybe we didn't need the rate limiting physics, or maybe we just need to crank up the Rodnina kinetics in our model. The answer is no. Let me show you what happens when we remove transport physics. Without it, sure, we do see the gap closes up, but we do not observe the speed up with growth rate that is well documented in the in vivo experiments. Physics is central to recover the qualitative behavior. And now to address the quantitative gap, what if we keep the physics and crank up Rodnina's kinetics threefold? Here we go. We can match the experiments quantitatively. However, that dramatic of an increase in vitro kinetics can't be justified. So we look elsewhere. Another venue to bridge the gap is to increase the biofidelity of our model, and that's what we do next. It is hypothesized that this interaction between L12 complex of ribosome and ternary complexes might lead to preloading of these ternary complexes and thus can reduce wait time. Jen Hoffman and Theo Yang, also from Zia Lab, incorporated these effects into our model using patchy surface attractions and studied the elongation process. And the result is very satisfying. The qualitative and quantitative agreement improves markedly. And we can now predict the absolute rate of elongation as well as the speed up. Before concluding, I'd like to thank Zia Lab members, Professor Zia and their collaborator, Professor Drew Endy, for their contributions. And for the conclusion, we see that the physical transport modulates protein synthesis rates and can explain the increasing ribosome productivity as the growth beckons. Here, using E. coli as a model system, we show the need to represent biomolecules explicitly and individually with physiological stoichiometry, and that was possible because of the massive data in the literature for E. coli, which was essential for faithful model building. And this is exactly what we need, an accelerated program to provide a similar data bank for Syn3A. And once we have such a data bank and build and validate our full-scale model for Syn3A minimal cell, we will work with our wonderful collaborators like Dr. John Glass and try answering specific biological questions and to connect these biological processes to transport physics. With this, I'd like to conclude my talk, and I'll be happy to answer any questions. Thank you for listening. Oops, sorry.
Vishal, that was terrific. I am dazzled by this work. I'm going to ask a slightly difficult question. Okay. So for years now, I have been working with the computational modelers at the University of Illinois and some other groups. And the work that I'm doing with them, so the way they are modeling the cell is at a different scale than the way that you and the Zia Lab is doing this. Mm-hmm. Can you contrast the two approaches and tell me what I'm likely to learn from one that I wouldn't learn from the other, and vice versa?
Yes, that is a great question. Okay, I'll start with one of the major differences here. And I think I briefly mentioned about this, how some models are just kinetic based. And from what I have seen in the work from the Illinois group, most of them are, they are wonderful work, of course, and they mostly focus on the kinetics of the reactions. They use the metabolic pathways, and they use reaction master equation and all of these wonderful methods to look into how they affect the synthesis of different biomolecules or these and like processes like transcription, etc. However, what is not accounted there, and I briefly mentioned it here in my talk, is that physical transport is often overlooked here because it is assumed that it is not really the rate limiting step, but we see that is not the case, and they could actually regulate the process, and that is something which we clearly see here, and that physical transport is something which I have not seen in other works. Yeah, and that is one of the main differences. And
Vishal, if you don't mind me jumping in to help you answer this question, I think one of the things that's really beautiful about the models that come from the Illinois group is it's a whole cell model, that it sort of gets the entire cell and can see what's going on for minutes. And one of our early approaches with this was to say, well, at that time, we didn't want to represent every single atom. You know, we didn't want to dump a bunch of atoms into a box and shake it up and hope for life to boot up, because that hasn't happened in billions of years. And we also felt like, well, let's not model the entire cytoplasm with our first model. Let's take one particularly charismatic process that occurs and understand that and study that. And when we make thousands and thousands of voxels to represent the entirety of the cytoplasm, that's one way to study it. But we knew that eventually we wanted to get to a whole cell model. And so that's where my group is working now, is we've got a whole cell confined. You can see sort of a section at the bottom left where there's a nucleoid for E. coli. And then, you know, the video is kind of jerky because of the internet, but these are very smooth models where physical motion arises naturally from first principles, F equals ma plus random fluctuating term. So our models are truly dynamic. The particles wander around physically in space, interact with each other electrostatically, entropically, through patchy attractions, and have chemical reactions. And so that's work that we haven't talked about today in the short time we had, which is the whole cell model that we're building. So I think always these two approaches are going to be complementary. And I think that we look forward in the future to gaining a lot of the metabolic and genetic information from other groups.